Welcome to the Genetics Podcast. My name is Patrick. I'm the co-founder and CEO of Sano Genetics. I'm here today with a really exciting guest. It's Dr. Yan Shao from the Sanger Institute, just outside of Cambridge. And I've actually known Dr. Shao for a while because we did our PhDs at the same place and actually rode the same bus into work some days. So I'm excited to have him on the show because he's recently published a very cool new paper that looks at the differences in the gut microbiome between newborn babies based on their delivery method, whether it's vaginal or C-section, as well as other parameters like whether they've taken antibiotics. And we also get a huge number of requests for people on the microbiome. I think it's probably the most requested topic area. So we're going to cover not just the new paper, but also a lot of interesting ground around differences in microbiome around the world and associations with disease. So very excited to have you on the show. Thanks for taking the time, Yan.
Thank you for the invitation. I'm glad to be here.
Great. Do you mind just giving us a quick intro to your new research paper, what you guys looked at, and how you ran the study?
Yeah, so in this new paper, we conducted the largest study of the human gut microbiota in very early life, and we recruited and sampled newborn babies in the UK of their microbiota via stool samples and performed shotgun metagenomic sequencing in the first month of life, longitudinally, and also for some of them, we follow up the microbiota later life. We found that among all the clinical covariates we collected, the mode of birth has the biggest impact on the microbiota, and specifically, the babies who are born by cesarean sections have a very low load of commensal bacteria, which is health-promoting bacteria, most of which are transmitted from their mothers. Whereas, on the other hand, by mixing that, they acquire a lot of bacteria from the hospital environment, which carry a lot of AMR, which is antimicrobial resistance and virulence genes. And we observe the differences throughout the first months of life and also at their infancy period. And we believe that by having this disturbed microbiota pattern, these C-section born babies are at the risk of future infections by carrying high load of pathogens.
Right. So to kind of recap the study, you looked at 600 infants across these three different hospitals, and you found differences in bacteria straight away based on the delivery method, antibiotics. And how long does it take for the bacteria to go from slightly different at the vaginal and C-section stage to kind of recover to what you'd expect if they were all delivered the same way?
Yeah, so you're right that we've seen the biggest difference in the earliest time point at day 4 in our study, and we consistently measure that significant differences, which is declining over time as you sort of enter the infancy period, which we also follow up their microbiota at around 9 months of age. But we still that differences is statistically significant, which is consistent with some of previous findings. But it does show that in the earlier period is the birth node that is the most important.
So is it too soon to tell, or do you know if these are likely to have other impacts later in life? Are you able to follow these families for the next 5, 10, 15 years to see if this disruption early in infancy has any long-term effects?
Yeah, so that's a very good question. So we don't know we don't have information that there's health differences. At least we know information we have. There's no health differences in these babies born by C-section or vaginal delivery, despite their huge differences in microbiota. Whereas the hypothesis that we want to following them in the later life, in say 5 years of age, is where normally the immune diseases develop, such as asthma and type 1 diabetes. That's we want to know whether the early life microbiota signature during the first month of life, which is a critical period of development for immune system as well. So what's the impact of the lack of commensal health-promoting bacteria transmitted from mothers in the C-section babies? What's the impact for them? Would they actually predispose them developing asthma in the later life, or would the presence of pathogens in these babies cause infections because they remain at risk, because as long as they persist in the gut microbiota, they can cause infections.
Right. So can you tell me a little bit more about microbiome science in general? How large are studies in general? I think for the kind of study you're doing, 600 people is a lot. But as you mentioned before, there are other studies going on in adults. How are these studies typically designed, and how do you collect samples? How do you analyze it? What are people typically looking for when they do them?
Yeah. So in this study, we studied 600 healthy babies in the UK, which is the largest of this type in the microbiome in terms of early life. Whereas in the adults, we're looking at the largest sample studies today, single study today is about 1,000 individuals.
Right.
So it's still big in the sense that a lot of samples will be collected, but it also, compared to human genetic studies, we're looking at over 1 million people
Right.
at some point, is probably still at very infancy period. It's because that we study microbiome by taking the stool samples, which might not be that easy to collect, convincing people to provide a stool sample for us to perform sequencing. Also, the cost of say performing shotgun metagenomic sequencing at the moment is still relatively high compared to doing genotyping in human blood. And also, sometimes our microbiome is much more dynamic. So it's not stable compared to human genome, where sometimes we need to take multiple samples. As the case in our study, we took longitudinal samplings of individuals, and we showed that by taking them, say, Monday and Friday, the microbiome could differ a lot in babies, whereas in adults, maybe there's small fluctuations.
Right.
So there are a lot of factors sort of important to be considered in designing a human microbiome study, because the samples depends on how it's being collected, how it's being stored, how do you extract the DNA, how do you store DNA, which sequencing technology you use. All these technical factors could contribute to the result. Yeah.
So you mentioned shotgun metagenomic sequencing. Is that the kind of state of the art right now? Because there's an older version that's been used in other. Do you mind just explaining the difference between those two technologies?
So the shotgun metagenomics is similar to we sequence the entire DNA of the community. So that's include not just bacteria, but also, say, if there are human DNA as well, we could sequence it as well, and also fungi, archaea, viruses. Whereas in the earlier phase of the human microbiome studies, most studies use what we call the marker gene based, which is based on a stickiness, our ribosomal RNA marker genes. So this is a conserved gene in all bacteria present in all prokaryotes, so archaea and archaea. So by sequencing that markers, you can detect what the bacteria likely is. It's like you perform which region of a chromosome, that in a human genome that gene is present. So it's limited by resolution, but it's much cheaper and much easier to study. But it provides you less information in terms of how accurate, because bacteria does different bacteria within one species could vary by their function. So right now, I think most studies microbiome are moving towards using shotgun metagenomics, which you can also find out new, complete novel bacteria, which actually is, we're still looking at the microbiome studies, very few bacteria we can culture to actually understand what are they. So
so there's a whole ecosystem in there, and if you just do the kind of cheap and simple way, you don't know what you're missing, right?
Yeah. So with shotgun metagenomics, for example, we now, by analyzing the entire human metagenomic data sets, we estimate that actually there's about 70% of bacteria species we have never studied in a lab before. So they only exist in sequences, but we haven't been able to culture it, which is another limitation of microbiome studies, that we don't know what bacteria is. We cannot culture it in the lab.
How much would it cost to do a single bacterial, if you want to do 1,000 bacterial metagenome sequences, about how much does it cost per sample?
So it depends on the technology and multiplexing, obviously. So it really depends. at Sanger, the sequencing center, it can be cheaper, but I don't think if you are from so university labs, it
Right.
can be, because after talking to people in conferences and community,
it can be really different.
Yeah, they sort of say that the reason they could not do circumvention genomics is limited by the sequencing cost.
Right. So, but at a kind of a large scale institution, would it be hundreds of dollars or thousands or tens?
I would say $100 is probably looking at not shallow sequencing depth, but like normal
depth.
Yeah.
Right. So for a study like yours to do 600 healthy babies, multiple time points is and it's expensive, right? So it's you have to have a research grant to do that kind of work.
Yeah, I guess that's why that it is quite. Also, sequencing is one aspect. A lot of effort and funding also spent on like collecting and recruiting patients and recording all the factors to be used. So I think that's sometimes with large cohort studies, I'm sure not just a microbiome, for human genetic studies, is a lot of hard work being done actually before the samples actually get collected.
Yeah, and you mentioned earlier that one of the challenges in the microbiome compared to human genetics, for example, is the constantly changing environment and different conditions that affect that. How do you handle that in these kind of studies? Do you have to collect all sorts of other information? How do you think about the difference between cause and effect?
So basically we try to collect and make sure all the as much metadata as possible, and make sure that the metadata are clean. I'm sure it is similar with other human genetics cohort. So in most microbiome studies so far, including us, we're looking at the association of factors with microbiome signatures. And so we get correlation of, say, factors and say C-section and increased load of pathogen carriage. But still, to be able to demonstrate causation, we need to go to the lab to demonstrate that by, say, using animal models. Can we replicate the findings? But that's, as I mentioned earlier, the limitation is that we cannot culture the bacteria, so we don't even have someone to start with. But the thing was, in our study, we be able to validate our sequencing based results by culturing the bacteria. So we make sure that when we see the pathogens carriage in babies is real, and they do carry a lot of air margins. So that's one way sort of trying to go further than sequencing detection, trying to say, can we validate it in the lab? And then the next step as trying to do now is trying to see, by using animal models, can we establish causation, say, do carriage or the missing of some commensal bacteria linked to some immune deficient phenotypes.
Right. So there's a lot of people are interested in how the microbiome relates to disease risk, and do you have any kind of broad overview? We hear often about immune conditions, bowel conditions like IBD, Crohn's colitis, but also autism is one that comes up frequently where people ask, is the microbiome associated? What's the current state? Do we know? Do we not know? Is it ongoing?
Yeah, so as you said, microbiome has been linked to almost all the diseases you can think of in human. And recently also, I think general of interest is linked to cancer. So sometimes, many cases, we find association with microbiota differences and sometimes particular type of bacteria. But in many cases, the studies cannot replicate or confirm each other. So they're reporting different microbiota markers. But that could also, that could be sort of indicating that there's microbiome differences across populations. So what the disease associated microbiome markers could be varied across, say, US population or Asian population or the African population. So I think at the moment, most microbiome studies are statistically underpowered. We is still at the stage of, say, looking at the p-value is something significant linked to a case control study or not. The markers still, at the moment, none of the diseases being associated with microbiome cannot establish causations. I think there's much more work to be done in the fields, much more. We need to sample more microbiome, and also in particular in different populations to compare what is actually. Firstly, I guess also depends on, can we define the healthy microbiome, which it seems to me that we don't have a consensus of what exactly is healthy microbiome at the moment.
Right, because it varies depending on diet, for example, right? You were mentioning before we started recording a couple of interesting examples based on the work you're doing now. How does diet affect it, or even geographical location?
Yeah, so diet is really important in terms of the adult studies. We're looking at, there's a striking difference in, say, high fiber. So fiber is important component in terms of bacteria like microbiota are specialized in digesting fiber and providing that to the human metabolic system. So there's a striking difference across population in terms of high fiber diets, low fiber diet, or high carb diets. So that's sort of, we know there's a strong selection on that in adults. In case of babies, we know that in the early life, there's a main diet differences whether you are being breastfed or not. So some babies are being exclusively breastfed during the first year of life. Some are only being breastfed for 1 month or completely not breastfed because the mothers have different conditions. But that has a strong impact in reaching the bacteria in the baby's guts. As I said earlier, the bifidobacterium is an important digester of human breast milk. And also, when we know that from literature, is that when you stop breast milking, also can shape when your microbiota is mature or not.
Right.
So diet definitely is consistently the most important factor in terms of shaping microbiota.
How about this this study you mentioned earlier of, they looked at recent immigrants to the US and I guess they analyzed their microbiome before or very soon after they emigrated, and then tracked it as they, I suppose, got onto the US fast food diet. Where did that study take place, and what did they find from that?
I think I might be wrong because I think it's a recent study in last year that's published in Cell, I think from Dan Knight's group. I might be wrong, from Minnesota. So basically, there's very cool study design in terms of the hypothesis that the changes in diet would change your microbiome. So first, you want to control the host. Basically, you're looking at recent immigrants arrived in the US from Vietnam, and so we know that they're gonna experience a shift in their diets because what they have been eating for their life before arriving in the US are very different from what they can get in the US. So they do the longitudinal sampling before and after, to right after, say, recent immigrants. They sample that and then they follow them throughout that first few years in the US, and then they compare the changes in microbiota, and they also measure the control group in the US, like the US nationals who have been living in the US in their whole life, and they find like a very big difference. As I mentioned earlier, the high carbs, high fats diet has a change to the microbiota. Yeah. Right,
and I suppose that change is probably associated with many of the chronic diseases that are prevalent in the US, for example.
Yeah, it's likely. And also the other, maybe the more prevalent use of antibiotics, more prevalent use of drugs, because we do know that the drugs can't, say, have an impact on specific microbiota bacteria or the whole microbiota. Basically, is that if you are a high antibiotic user, you are basically dropping, as Trevor, my supervisor, I like to use an analogy, dropping a bomb to the microbiota, completely eradicate all the bacteria, and then all the microbial resistant bacteria can sort of survive.
Right.
Like weeds, they're always easy to grow, which we see in the C-section born bacteria babies. Yeah.
Right. So I guess it sounds like the current understanding, and I understand why now it's such a complex field, is you've got some steady state microbiome, but it's being influenced by your diet. Yeah. If you have a condition like IBD or ulcerative colitis or Crohn's, that's affecting the way your immune system interacts with your gut. Anyways, your personal genetics might influence the way your immune system interacts with your gut. Where you live impacts this. And so it's constantly changing. And I guess this makes it really difficult to figure out, does the diet change the microbiome, which in turn interacts with the immune system, because you've got this complex system it's no one factor essentially that's doing everything, right? It's some complex combination of multiple things.
Yeah, and also we don't want to forget about the human genetics role. So as always talking a lot about an environmental role, we do know that human genetics play a minor role in the microbiota. So that's the studies looking at a cool study is basically they're looking at the Israeli populations, and they know that where they immigrated from,
right.
different parts of the world. So they have different genetic backgrounds. And then once they settle here, they have sort of relatively controlled diets. So
right.
then there's a cool study looking at what's the contribution of genetics and contribution by genotyping the participants, and what's the contribution of diets or other environmental factors. So they showed actually genetics only play, I think, around 15% of the variation, only 15% variation in microbiota compared to the rest of our environment. And also in like the twins studies, like twins UK studies, so looking at twins but with different microbiota, and then we can sort of measure what's the inherited microbiota that are shared by twins, or they're not shared by twins. So there are actually very few bacteria you find in the twins microbiota, even though they share the same growing environments or relatively similar diet.
Right. Yeah. Those studies always fast. So in that case, would the twins be, would they be twins that are separated at birth? So they have different environments, but same genetics, for example?
Yeah, I got, so in that case, they definitely more similar microbiota compared to the microbiota differences with random people, stranger. Right. But even though you would assume that with, as the context of human genetics, they should be
very similar,
close to identical. Yeah, but like with microbiota, you found actually quite different, and very few bacteria actually shared between them. But if they are, but that's because they not live together anymore. But in case of, we know, like in family studies, you share more microbiota with who you live with, or that could be a family with a partner, or it could be with student dormitories in universities. You probably share the same microbiota. Then one person, because you share the kitchen, you sometimes share the same cutlery, same the water bottles, glasses. Yeah, so because microbiota can spread between people, and what we know so far before is that the bacteria are probably associated with pathogens, infections, but actually majority of bacteria and the thousands of species, the trillions of bacteria strains found in our guts, they are mostly commensal. They are sometimes related to promoting health. So by spreading microbiota, you are also we believe that spread health as well.
Right.
Yeah. So that's sort of like very, 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 very interesting field.
Yeah, I mean, we tend to think of them. I feel like this perception is changing, but we tend to think of bacteria as the bad guys, but actually probably the vast majority of them are living symbiotically with us and doing good things like digesting breast milk and helping break down food and that sort of thing.
Yeah, exactly.
Yeah, that's fascinating. So where do, just this week, the direct consumer company Ubiome, who was selling at-home microbiome tests, declared bankruptcy because I think they had some issues they had some legal issues where they had bad billing practices. But I think also maybe it was a little early or premature for this kind of analysis for a consumer. Do you have any thoughts on how does direct to consumer testing and research, what can someone learn from doing this at home? Is it kind of primarily a curiosity at this point, or are there some areas where actually, if you committed to paying £100 a month to do a microbiome test, you might learn something? I see you laughing and smiling. I know it's a hard question. Yeah.
So I guess this come back to the point that how developing potential diagnostics in the microbiome field. But again, microbiome is dynamic. It can change. So let's say we have a test for microbiome and there's much better resolution than what's currently offered by, or used to be offered by Ubiome at 16S, or basically give you a very high level overview of microbiome. So let's say we have best test, but then even though, does it mean that test is your microbiome? Does it mean that microbiome change a lot and you need to do microbiome tests every day? Or even during the day, probably it fluctuates as well. So probably I don't think there's a current like framework in terms of
Right.
how do we go about from microbiome in terms of establishing a biomarker, because we still lack of a lot of fundamental understanding of the causal effect between microbiome to ending disease. We roughly know, say, having a healthy high fiber diet probably promotes good bacteria in your gut, but we don't know whether that's going to prevent you from getting any disease or not.
Right.
I think right now I wouldn't discourage people who taking it. It's for participating in scientific research or for the scientific curiosity, understanding what microbiome is. But I wouldn't rely on that to give any sort of diagnostic or prediction of any disease outcome at the moment, because we're still very far away from reaching that point. I guess that also echoes with the human genetics.
Yeah, exactly. I think that is good advice. I was exactly going to say that in human genetics in general, there are some cases where it might tell you something very useful that you don't know, but the vast majority of those cases are still in the clinic. It's coming from doctors, not in, but again, people who are interested in this and want to contribute to research, like you say, and and maybe learn something by actually being able to have the data themselves and play around with it, then it totally makes sense. It's amazing to think about that, though. Someday we'll have I'm sure someone will invent a device that will allow us to constantly monitor the microbiome in combination with activity and diet and information about our genetics that could help unpick this very complex picture.
Yeah, actually, there's a recent startup on developing using toilets to measure sampling microbiome.
That makes total sense.
I think that's going to be the future in terms of it's going to be much easier to collect samples, and in terms of direct to analysis and reports. I think also in say the developing countries where it's not that easy to acquire stool sample and to put in a tube and strip it in dry ice, those types of very difficult conditions. I think it's gonna be also using how can we potential of using microbiome to as a surveillance of say pathogen carriage in the guts in say, epidemic areas in terms of the carriage, because a lot of gastrointestinal infections like cholera and E. coli infections, something like that. Basically, by sampling healthy people, we found that actually they are present in healthy people as well. It's just that they're not causing diseases. So we can potentially use that to sort of monitor the presence of the high risk bacterial strains like cholera or salmonella in the human gut, and can we probably inform the infection prevention strategies.
Yeah, that's fascinating. It could be an early warning system of some sort, right? Where you like you say, you pick it up at low levels in healthy people and can flag it up as increasing in this area, for example.
Yeah, or can we inform, say, the antibiotics usage? If the person carries a high load of AMR carrying bacteria, does it mean that it might enrich your AMR load in the gut microbiome, because that can transfer, AMR genes can transfer between healthy bacteria and pathogen bacteria?
So the issue of antimicrobial resistance, is it getting better or is it getting worse? I know for the last 5 years or so, it has been near the top of a lot of the World Health Organization's agenda. Is it getting better, or is that kind of, are we headed for a big mess when it comes to antibiotic resistance?
I certainly think that there is that trend in terms of that AMR is becoming a major issue in public health. But I think by understanding more and more about microbiota, even a healthy microbiota carry AMR genes. The key question is that are we running out of antibiotics? Basically, there's lack of new antibiotics that can kill the bacteria with AMR genes, because what we have so far, more and more bacteria are resistant to the last line antibiotics, which means that if patient got infected, all the antibiotics available cannot kill the bacteria. That's the major issue now, because they develop resistance. There's a lack of investments in research on the new antibiotics, because there's always this evolutionary arms race between bacteria and say drugs. They can always human can always develop new antibiotics, but the speed is not as fast as bacteria can
develop resistance to it.
Yeah.
Wow. Yeah. So if any grant funders out there listening, this should put this, I know the Wellcome Trust, this is pretty high near the top of their list, right?
Yeah, because there's lack of interest from the pharmaceutical companies, because there's less profit. It's normally targeting towards developing countries. It's difficult to sell at a higher price, or it cannot justify the investment into R&D. So I think this definitely requires more public investments from government or health agencies in terms of encouraging more research into new antibiotics. Otherwise, we might actually looking at running out of antibiotics that can treat future infections. Yeah.
Right. Yeah. So thinking about the future in general and the future of the microbiome research, what do you think are the most exciting things going forward in the next, say, 10, 15 years of what we should be working on or what we have to look forward to?
Yeah, I think this definitely, as I said earlier, it's very still in infancy, that we only know so far because we undersampled a lot. And mainly what we know so far, microbiome are in the large studies in US, in Europe, and in China. Globally, we don't have any information on what's the microbiome of, say, the African population, which is incredibly diverse compared to European, Chinese, American populations. And also, what's the different lifestyle changes in the microbiome? And again, we're trying to tap into discovering new bacteria in humans. So we only right now working with handful of bacteria in the lab in terms of these are common bacteria found in human gut. Whereas in sequence analysis, we found actually majority of bacteria have never been cultured or studied before. So I think the next phase in microbiome research will be trying to understand what are these bacteria doing by first isolating them, to find them and study more in detail, because that could actually fill the gaps in terms of how different microbiota between people can associate with different health and phenotypes.
Yeah, no, that makes total sense. Like without being able to culture it or understand what it's doing, then you really are missing that next link, because you can observe a relationship or correlation between some change and how it relates to disease, but if you can't prove that cause, then it limits the usefulness, doesn't it?
So it's like you don't have a complete human genome, and right now we realize that we only studying on one chromosome or two chromosomes of the entire human genome and compare the whole thing with each other. So that's the current state of microbiome we only know so far. And also that's with the Western populations or Chinese populations. We don't know anything about, say, what happens in other countries we haven't sampled. So what we are doing in our group at the moment, we're trying to initiate new collaborations with low middle income countries in Africa, in Asia, and in Australia, trying to look at those undersampled populations, what's the microbiome differences, and how can we improve our knowledge in understanding the microbiome association with health and diseases.
Right, that's amazing. Well, if people want to keep in touch with your work, follow you, read the next exciting paper, where can I do that? I know you're on Twitter. What's your Twitter handle?
Yan Shao with 2 underscore in between.
Okay, 2. Don't forget that second underscore. So YAN __ SHAO. And you're in the Lolly group at the Sanger Institute. So there's more information on the website as well.
Yeah.
Great. Well, wonderful. Thanks so much for coming on the show. It was amazing. I learned a ton. I think I'll have to do some more background reading and try to dig into some of these issues. It's my mind is blown by the complexity of it all. The time, diet, disease state, healthy state, all of these things are going to impact it, which just makes it really painful to understand what's going on. So I think we salute you for your work in embracing the difficult challenge that it presents.
Okay, thank you. I really enjoyed the discussion.
Yeah, my pleasure.